Welcome, everyone. My name is Sally Syme, and I'm the Director of Public Programs for Audubon's Corkscrew Swamp Sanctuary. And we're so happy that you're here with us today for our Lunch and Learn. And today we have our speaker, Courtney Witcher, who is a PhD student at Florida State University, here to present her program, Biofluorescence. But we'll get started in a moment. But first, we'd like to remind you, if I can get my computer screen to work, would be great. It's not working, unfortunately. But anyway, I just wanted to remind you that since we have so many people signed on, we're not actually having everyone share their photo or have any audio function. But at the bottom of your screen, there is a chat button. If you have any questions during the program or afterwards, you can type your questions into the chat button part of your screen, and we'll address those at the end of the presentation. A recording of this webinar will be available both on our website and also on our YouTube channel within a few days, if you'd like to watch it again or refer it to any of your friends. So now we'll let Courtney get started with her program.
Great. Let me just share my screen really quick. All right, so thank you so much, Sally, for the introduction. As Sally mentioned, I am a PhD student at Florida State University, and today I'm going to talk to you about finding fluorescence and discovering this glowing world that surrounds us. So I want to begin by talking about this photo that I had on that first slide. So this is an artist's interpretation of exoplanet biofluorescent biosphere. And this is based off of research done at the Carl Sagan Institute on life adapting to young stars with large UV fluxes. So our first targets in the search for signs of life in the universe are orbiting near what astroscientists call M-type stars. And these young M stars tend to be very active, and they flare frequently and cause UV fluxes on the surfaces of planets. And so these strong UV flares can be biologically harmful, as we've all likely experienced with a bad sunburn from a vacation to the beach. But one biological photoprotection method, or method to protect life from UV rays, is called biofluorescence. What biofluorescence does is it upshifts the UV wavelengths to longer, safer wavelengths. And the scientists whose research this poster is based on suggested that we could use this biofluorescent biosphere signature to detect and reveal life on other planets. But what I hope to show you today is that we're already living on a glowing biofluorescent world. This isn't just a potential way to search for life on other planets or a science fiction phenomenon. It's embedded in the systems around us. So we're already living on a glowing planet. So let's explore this glowing planet that we live on. What's this thing called biofluorescence that I keep mentioning? In the most simple terms, biofluorescence is glowing life, or glowing species. But there are two ways that species can glow. These are called biofluorescence and bioluminescence. You're likely most familiar with bioluminescence, so let's discuss that one first. Bioluminescence is the biological production of light. It's likely that many of you have witnessed a firefly or lightning bug in your backyard, or remember the glowing lure hanging in front of the anglerfish's face in the Finding Nemo movie, or maybe you've been able to paddle or swim through the ocean while bioluminescent plankton are out. These organisms all glow via bioluminescence. Here we have a depiction of the bioluminescent tree of life with representations of many of these glowing species. So from bacteria to fungi to fish to worms and insects, squid and crabs, bioluminescence is present in many different types of organisms. But the interesting part is they all glow using the same exact mechanism. With a special molecule called luciferin and some oxygen, all of these species glow in the same way. So I want you to take a minute to think about that. Look at the diversity of bioluminescent species on this tree. A single-celled algae and the 46-foot-long colossal squid glow in the same exact way using this molecule right here. Pretty incredible, I'd say. So we have all these glowing species that are bioluminescent, but what's biofluorescence? And how do biofluorescent species glow? Have you ever been to a bowling alley or a haunted house where your shirt and your shoelaces glow bright under the black lights? That's fluorescence. And when living organisms do this, it's called biofluorescence. So biofluorescence is another way that species can glow, but instead of producing their own light, they absorb light and they re-emit it in another color. And some species can fluoresce in wavelengths outside of the range of colors that humans can see. So it can change species that look like this in our eyes to looking like this. Now let's talk a little bit about how biofluorescence works and the science behind it so we can really understand what's happening. Biofluorescence is found in species across the tree of life, similarly to bioluminescence. We see biofluorescence in over 180 species of fish, hundreds of species of corals, in jellyfish, in sharks, in rays, in sea turtles, in penguin beaks, in parrot feathers, and owl wings. Every species of scorpion is biofluorescent. We have biofluorescent spiders and butterflies and beetles and dragonflies and cockroaches, and nearly all plants are biofluorescent, as chlorophyll is. And then also we find biofluorescence in over 35 amphibian species, frogs, salamanders, and caecilians, and in many species of reptiles, from snakes to lizards to turtles. Now, I want to revisit these images that I just showed you for a minute. Look at the differences in biofluorescence. Each organism glows in a different color and a different pattern, and these differences in fluorescence are related to its various functions in each species. Some of the reasons scientists have found for biofluorescence are it can act as a signal of condition, it can act in attraction, interspecies recognition, it can be a warning to predators, it can help species camouflage, it can be used as a lure for photoprotection, like we talked about in that planet at the beginning of the talk, or for symbiosis or aggression. And I'm going to walk through a few of these examples so you can get a good idea of what this means. First, many flowers have UV patterns, specifically this bullseye pattern that we see pictured here. And it's predicted that this pattern is used by pollinators like bees that can see in the UV range to focus in on areas for pollination. And the UV bullseye pattern makes it easier for the pollinators to find the reward of nectar and therefore aids in pollination. Next, here we have a picture of a red fluorescing scorpionfish perched on red fluorescing algae on the top, and then on the bottom, a green fluorescing unipterid fish near a green fluorescing coral head. As you can see, the fish are located near organisms in their environment that are fluorescing the same color as they are, likely aiding in their camouflage. Now, this third example, we have jumping spider fluorescence. The top image is of male spiders on the left and females on the right in natural light, and then the bottom image is the same spiders under UV light. And researchers found a decrease in the number of courtship responses when the UV wavelengths were removed. So males were uninterested in females that lacked fluorescence, and females were uninterested in males that lacked fluorescence. Now, here's an example a little closer to home. Fluorescence isn't always connected to a communication signal. It can be a product of cell aging. We see this in the ripening of fruits, as you can see pictured here with the bananas, but also in the aging of mammal tissue, like heart, liver, kidney tissues, that are in our own bodies. So we see this in the image on the bottom right, from a 4-month-old and an 18-month-old rat. The white fluorescence in the brain increases with age. So theoretically, you could take a black light and use the emitted fluorescence to tell you the age of both your fruit and your body. Now let's talk about an example much further from home. We're going to look into the deep sea. So this is a siphonophore, which is a colonial hydrozoan. It's related to corals and jellyfish, and it has a red fluorescent lure at the end of its tentacles to catch prey. So because siphonophores are mostly deep sea creatures, there's not much ambient blue light to excite the fluorescence. Hence, they have to produce it themselves via bioluminescence. So siphonophores are both bioluminescent and biofluorescent. The tentacles are blue luminescent and only produce red biofluorescence once they mature, when they're then hypothesized to use it as a lure seen by the action in this video. So this might remind you of the bioluminescent lure of the deep sea anglerfish. The depths of the ocean are a dark and lonely place, making communication via light very valuable. Hence, this is likely why we see so much bioluminescence and biofluorescence in marine species. So here we've had examples of biofluorescence used to communicate between species with the flowers and the pollinators, within species with the jumping spiders, for camouflage in reef systems, as a sign of condition in fruit, and as a lure in siphonophores. Now I want to mention one special example that has really important implications. So this is the crystal jellyfish. Again, it is both bioluminescent and biofluorescent. It produces a blue glow via bioluminescence, and then the light from that blue glow excites a green fluorescent protein that causes green biofluorescence. And research on this green fluorescent protein, or GFP, as you may have heard of it, garnered the 2008 Nobel Prize in Chemistry, which has been now used extensively in the medical field as a marker to identify and examine biological processes through a method called fluorescence microscopy. Because it's heritable, it'll show continued expression of the cells and tissues it's expressed in, and it doesn't interfere with any biological processes. So here's a real-life medical implication of studying this biofluorescence. Now let's talk a little bit about how biofluorescence works. So biofluorescence is the result of natural fluorophores, which are just chemicals that fluoresce, and there are many of these, which they're all organic chemicals, as you can see by the similarities of the rings in these few structures that I've shown, and they all have their own fluorescent emission wavelength properties. For example, one of these names you might recognize that I have up here is chlorophyll. As you know, that's what makes plants green and allows them to photosynthesize, and it's also a fluorophore. Now, there are hundreds of fluorophores, so each fluorescent organism glows in a slightly different way. And biofluorescence is the result of the absorbed light being re-emitted at a longer wavelength from these fluorophores. And the wavelength of light determines if we can see it and what color it appears as. So as you may know, light acts as a wave, and each color has a different wavelength, which is the measurement between the two peaks of the wave. So the wavelength of red is longer than the wavelength of purple. Here, the wavelengths of the light visible to humans are also labeled in the bottom left, and some organisms can also see wavelengths in the ultraviolet range. Now, fluorescence shifts this wavelength to a new color, and this is called the Stokes shift, and this diagram on the right shows you what that is. So for example, it would absorb this blue wavelength and then shift it to a longer wavelength, emitting this green-yellow color. So let's put that all together in an example. We know that plants have chlorophyll, which makes them green. We just learned that chlorophyll is a fluorophore, which has a peak excitation wavelength around 480 and an emission wavelength around 700 nanometers. And so we can see that if we shine this blue 480 nanometer light onto a green plant, that it will re-emit that light at a longer wavelength at around 700 nanometers in this red color. So your green plant will look red. And the amount of and wavelength of the red fluorescence of plants has also been correlated with environmental stress conditions and is now used as a tool to gain information about pollutant and stress effects on plant health. So what I really want you to take away is that biofluorescence can reveal the unseen. Here we have just a few pictures. On the left, it looks like normal sand, and then you can see that there's a hermit crabs and shrimp in the sand that are exposed when you look in fluorescence. Similarly, on the right, you can now see the hornworms on a tomato plant. So we live in this glowing fluorescent world that we just don't always have the ability to see. Now let's talk a bit about the biofluorescent organisms that I study, frogs. The first biofluorescent frog was discovered just 3 years ago in 2017. It was discovered on accident when someone mistakenly shone a black light on a frog. And because it was just discovered, we still don't know why frogs are biofluorescent, which is where my research comes in. So since 2017, researchers have found biofluorescence in over 35 amphibian species and across the 3 main groups of amphibians, frogs, salamanders, and the rare burrowing amphibian caecilians. Personally, I've discovered fluorescence in 5 genera of frogs and about 10 local species now, and documented the first notes of intraspecific variation in fluorescence. So this means that individuals of the same species don't always have the same amount of fluorescence or the same fluorescent pattern. And this discovery provides more evidence that biofluorescence could be playing a role in frog ecology and evolution and might not be just a byproduct, which led to my research on trying to understand the role of biofluorescence in frogs. We're also seeing differences in the excitation and emission spectra of amphibian species, which you can see from the range in fluorescence color from greens to blues in these photos. So now let's look at frog biofluorescence a little more closely on the evidence that we have for why it might be playing a role. But remember that there are hundreds of different mechanisms of biofluorescence. So different frog species may have different fluorophores underlying their fluorescence. We only know the mechanism in one frog species. And again, this adds to the evidence that it might be playing a role in some species, but not in others, perhaps. Let's look at amphibian vision, because biofluorescence can't be playing a role if frogs can't see it. So rods help organisms see in dim light, and cones help them see in bright light. And having an extra rod, this blue sensitive rod, helps them better see in dim light. I just saw that Maureen raised her hand. Do you have a question I can answer? Oops. I don't know if I'm able to see the chat while I'm here.
I can't see that she raised her hand. We'll just save all the questions for the end.
Okay, no problem.
If you have questions, just type it into the chat box.
Great. Okay, so as I was saying, these rods help organisms see in dim light and cones help them see in bright light. So having an extra rod, this blue sensitive rods that frogs have, helps them to better see in dim light. But the most exciting part of this is how frog biofluorescence excitation and emission spectra interact and line up with these rod sensitivities. So biofluorescence pushes the emission wavelength to the most sensitive range of the green sensitive rods. And because they're more green sensitive than blue sensitive rods, and the emission is pushed to the wavelength of high sensitivity in both the green sensitive rods and even the red sensitive cone, fluorescence is making frogs easier to see by other frogs. And this is in dim light, which is when frogs are really active, specifically when they're mating. So perhaps frogs are using biofluorescence to communicate with potential mates. Now here's some evidence that I found locally to help test this hypothesis. Here we have Hylasquarella, which has been found to use this white stripe that they have as a secondary female preference cue. I also found that this white stripe is fluorescent, so perhaps it's the fluorescence driving this female preference and not the white color of the stripe. Now let's look at predator vision. Many predators of frogs can also see in this near UV range that frogs are biofluorescing in. Some birds have an extra cone with sensitivity in the UV range, as seen by the absorbance spectra on the right. And additionally, while owls, a main predator of many frog species, don't have this cone, they have UV transmitting eye media, allowing them some sensitivity in the UV ranges. So there's reason to believe that bird predators are likely seeing frog biofluorescence. Now what about snakes, another main predator of frogs? While they use smell for most prey hunting, it's predicted that many of the nocturnal species of the group of snakes, containing over 80% of snake species, have UV-sensitive optic ranges, based on their most recent common ancestor having this trait as well. So it's likely they can see it too. And then mammal predators, some romanids and marsupials have a fourth cone like birds, and other mammal predators like ferrets have lenses that allow the UV light through, similar to owls. Now I do want to point out that the human lens, as you can see on this graph, block out most of the light between 300 and 400 nanometers, which is the range of wavelengths that a lot of biofluorescence, including frog biofluorescence, is in. And this is why we can't see most of the biofluorescence surrounding us, at least without help from special lights and UV filter glasses. So we know predators of amphibians can see in the correct range to see their biofluorescence, but what does this mean? Biofluorescence could help frogs blend into their fluorescent-filled background, helping them camouflage and evade predators, as patchy fluorescence was found to be correlated with camouflage function in fish, and we now have discovered patchy fluorescence in frogs. This might provide evidence for testing that hypothesis. Alternatively, biofluorescence may make frogs more visible to predators, but may act to deter them, similar to how bright colors of poison dart frogs and coral snakes warn predators of their toxicity. Specifically, rodent predators of the polka dot tree frog, which was that first biofluorescent frog discovered, are deterred by the irritating secretions of the frog, and these secretions are also fluorescent, as pictured here on the bottom. So perhaps biofluorescence is used as a warning mechanism to predators. And now all of this information leads to my main research focus. I'm examining the role of biofluorescence in frogs, specifically in mate choice and predation. And there are 3 steps to this. So first I have to describe the variation, then I do female preference tests, and then some predator encounter experiments, and then look at the evolution overall. So first, to understand the function of biofluorescence in frogs, we have to understand its variation within and across species. To begin to understand the variation, I catch frogs, shine a light on them at the excitation rate that we found other amphibian biofluorescents at, and record notes about the color, pattern, distribution of the biofluorescence, and about the environment, time of day, breeding season, etc. And then I collect the emission spectra using a spectrometer, which is pictured here on the right. And this gives me the excitation and emission spectra by reading the wavelengths of light emitted off of the frog. So I can then know the peak emission wavelength and the intensity of the fluorescence and use these parameters to compare biofluorescence across individuals and across species. And so this work helps me decide which species I should focus on for the mate choice and predation experiments. I can see if fluorescence is in a biologically important place, like the vocal sac, or if wavelengths match to predator visual sensitivities. I've also been collaborating with other institutions like the Detroit Zoo, the Coastal Plains Institute, the QCAS Museum in Ecuador to test a wider range of amphibians that I wouldn't have access to otherwise. So then once I quantify this biofluorescence, I want to see if it plays a role in mate choice. And to do this, I set up female preference tests where I place 2 males in an arena, I block the fluorescence of 1 male using different lights or filters, and then see if the female chooses the male with the fluorescence or the male without the fluorescence. And then I can repeat these tests with different fluorescent patterns or species or acoustic calls to start untangling if biofluorescence is important in species recognition and how it interacts as a cue with acoustic signals. I'm also collecting blood and DNA samples from individuals with more and less biofluorescence to see if there's a hormonal or genetic difference correlated with biofluorescence. So perhaps a reproductive hormone like testosterone is correlated with more biofluorescence. And then to test the effects of predators, I'm making and placing wax model frogs. I paint some of them with fluorescent patterns and leave some of them normal. And I put them out and I look at the interactions with predators by examining bite marks, claw marks, and by using cameras. And this helps us to determine if biofluorescence deters predators or attracts predators and how it might be affecting different predators differently. And then finally, I can use all of this information to start to understand if and how biofluorescence is affecting the evolution of amphibian species. So I can map biofluorescent traits like excitation wavelength, intensity, pattern, color, function onto the relationship tree of frogs and see if these traits correlate with an increase in speciation or other evolutionary processes. So here's an example of mapping different colors of fish biofluorescence onto their tree. Now the findings of this research are not only going to better help us understand how frogs communicate and interact with their environment, but this knowledge can help us to better be able to protect and conserve them and their ecosystems, especially amphibian species, which are a huge indicator species that provide insight into the health of our environments. Similarly to green fluorescent protein applications, understanding other fluorophore chemicals can provide uses in the medical field for microscopic tracking and imaging as well. Now, I love my research, but science isn't as useful unless it's shared, and I can't possibly study all of the wonderful aspects of fluorescence myself. So I've created a way for you to get involved in finding fluorescence. There's still so much we don't know about fluorescence, specifically because we haven't taken the time to shine a UV light on every species. And so finding fluorescence is a research that I created to help with this problem. The goals of this research are to teach about biofluorescence, get people involved and excited about making discoveries, and to document the presence or absence of biofluorescence in the vast number of species across the world, and make this data accessible to scientists of all fields. So to reach these goals, finding fluorescence utilizes 2 different parts, the citizen science project and some educational resources. So first, if you remember the beginning portion of this talk, many species of plants and animals glow. And with an inexpensive black light and UV filter glasses, you can find biofluorescent plants, insects, amphibians, reptiles, fungi, fish, and much more in your own backyard. And these are all images uploaded to the Finding Fluorescent database by citizen scientists just like you, normal people in their backyard with just an inexpensive black light and filter glasses. So here's a quick video to show you what this citizen science looks like in real time. If you look at the top right corner, you can see now here's the UV light, and we're going to put the filter glasses on in a second. You can see the green leaves automatically turn red, if you remember from that chlorophyll fluorescence. And then as the spider moves, you can see that he has a little bit of green fluorescence underneath him as well. So now the data that's being generated by this resource is extremely valuable. Recently, I spoke with the director of herpetology at the University of Michigan Museum of Zoology, who brought his black light into the collections and found fluorescent tubercles in a new species of chameleon and sent me this photo pictured in the bottom middle. And then we also had a photo of a biofluorescence in a sand crab at the top right, or mole crab you might know it as, from New Jersey posted. And these are both the first documentation of fluorescence in these species. So it's pretty incredible that you can make a huge scientific discovery having fun with just a black light in your own backyard. Now, that being said, one of the scientifically important features of the Finding Fluorescent resource is this database that's created from the data that citizen scientists upload. So this data can be accessed and downloaded for free by anyone and can help scientists better understand the diversity of biofluorescence across all taxa and across all geographies. It can also help us better understand how biofluorescence is affected by geography, seasons, and other environmental factors. And people can easily upload whatever they find through a phone app or an online web page. Now, the second feature of the Finding Fluorescence resource is education. So biofluorescence provides a really unique opportunity to teach and learn about the three main topics of science all in one lesson. So you can learn about biology, chemistry, and physics all in biofluorescence. And so Finding Fluorescence website has free downloadable worksheets available for use in the classroom at home, which connects this fun activity of taking a black light out in your backyard or the area around your school to the science behind it. And all of these worksheets and the website are now available in both English and Spanish. I also have Finding Fluorescence kits available for free rental so that classrooms or summer camps or scout troops can request this if they don't have funds to purchase the equipment needed to the activity themselves. They can rent it out for a period of time, utilize it, and then send it back. Now here's the website, and I'm just gonna take you through it really quickly. So this is the homepage that just describes what Finding Fluorescence is. We have an About Biofluorescence tab, which talks about the difference between biofluorescence and bioluminescence, and then goes into a little bit more detail about the biology, chemistry, and physics behind biofluorescence, so you can better understand what you're looking at before you go out, or use this to help your class understand. Under the Get Started tab, we have information on data collection, classroom resources, and more. So it tells you where you can look for biofluorescence, where it's present, how to identify it. It gives you some links to really expensive black lights. You can get one for about $3 off of Amazon. It shows you how to tell what's fluorescence and what's just reflectance. Here's where you can download those worksheets. They're available. Here's the biology one. You can download these, do this classroom with your kids at home, with a summer group, or any other group that would like to. There's also worksheets for chemistry, physics, and just a lab. Here's where you can request the Finding Fluorescence kits. And then as you go to the next page, here you can view all of the citizen science uploads that people have posted. So you can also view this in a map form where you can zoom into your hometown and see if anyone has found fluorescence near you. For example, you can click on a specific observation. It shows you what they found. Yes, they found fluorescence, they found it all over, and then it shows you photos of that organism that the citizen scientist uploaded in both UV light and in natural light. You can upload data yourself to the web form or on the app, and then there's a contact page if you want to request one of the kits, you have any suggestions on what you would like to see out of this resource as well. And like I said, this is also all available in Spanish too at the moment. So in this talk, we've seen that there's a bioluminescent world that's glowing in front of our eyes that we can see. But there's also a biofluorescent world, and that's often hidden from human sight. But we live in a glowing world, and you have the ability to discover it. So if you got one thing out of this talk today, I hope it's that you also live in this beautiful biofluorescent world. And I am happy to take any questions. I also have my contact information up here, that website link. This is a little QR code. You should be able to hold your phone up to the laptop screen and go right to that website if you don't want to type it all in. Thank you so much.
Okay, thank you so much, Courtney. That was all very interesting and exciting that we can find these things right in our own backyards. I can't wait to go. Exactly. So we do have a few questions. One of them was, you mentioned that you found 10 local species that were biofluorescent, where they were all frogs, right? What were some
Yeah,
of the other ones?
yeah, I've been testing frogs specifically for my own research, but for a lot of this finding fluorescence resource, I've been going out with my family too because I'm interested in everything else that's fluorescent. So in species besides frogs, we found a fluorescent mosquito, lots of different plants and insects, lots of caterpillars and spiders and invertebrates. But there was also that sand crab, and all of all of those findings you can see on the Finding Fluorescence website. If you go to that view map tab, you can look through everyone's pictures, and there's even more. I took that video a few weeks ago, so there's even more uploaded now about some of the stuff that people have found that's not just me too.
Cool, that's great. Okay, so someone else had a question. What use do deep sea animals have for fluorescence? That they would imagine there's very little light that reaches that depth.
Yeah, that's a great point. So the reason that they can still do it even though there's little light, a lot of times is because they're also bioluminescent. So they produce their own light and then they will take that light that they emit and use it for fluorescence and re-emit it. So that's how they do it. But a few of the uses, we found it's used in lures, like I showed you the video of, but a lot of it is for species recognition they're finding too. If you're in this vast, dark place really deep and there are very few of your species roaming around, it's nice to have really distinguishing features that you can see that are really different from your background, from the dark background. So this light is going to attract not just other individuals of your own species, but potentially other prey too. And so those are the two main reasons we're seeing it in deep sea creatures.
Okay, thank you. That's great. Someone wondered how much chemistry you studied in undergraduate and graduate school to be able to do something like this.
Yeah, I studied a good amount of chemistry in undergrad. There was a point where I was thinking about doing a biochemistry major instead, but a lot of this is utilizing collaborators. So I have some really great collaborators down in South America who are on the forefront of understanding this biofluorescence and the chemical mechanisms. And so we work together where I can test some of the behavioral stuff, give them skin samples, they can try to figure out some of the chemical underlying mechanisms, and then we can get a fuller picture together.
Cool. Very cool. And another question was, are there any research findings that you can share with us or any links to publications? Are they on that website or?
They're not on there are some on the website right now. I can send more out or I'll put them on the website too. But if you search biofluorescence in whatever species you're interested in, you'll have a whole list of things that come up. One of the resources that I have, I believe, linked on the website is a whole review paper that looks at biofluorescence in all of these species across many different taxa, and it cites a bunch of other scientific studies as well. And so, yeah, there are definitely many resources, and feel free to contact me too. You can email me anytime, and I would be happy to send along even more.
Okay, wonderful. That's great. And I'm just wondering if you're doing any more presentations or how you're trying to get the word out to people all over the place.
Yeah, I don't have any set in the near future, but I'm trying to collaborate with local or not local education resources. So if you know any classrooms or teachers that would like to utilize this resource and it'd be helpful for me to come and do a little quick presentation, I'd be happy to. Any summer camps, museums, places like this, sanctuaries, I'd be happy. If you have any connections, please let me know, or if you'd like me to present anywhere. But either way, you can go to this website and utilize those resources for free, and let me know what else you'd like to see, because I'm trying to constantly make it better for everyone.
Okay, great. All right, well, let me just check and make sure if anyone else has any questions before we go. Okay, it looks like that was the last question. So, but thank you so much, Courtney. We really learned a lot. It was a very interesting presentation, and hopefully you got a lot of us excited. I know I am, to get out there and look for some of these things.
Great. Yeah, thank you so much for letting me present, and I hope you all enjoyed.
Okay. Thank you. Have a good day, everyone.